Today I'm going to give you an introduction about CRISPR toolkit development for natural products discovery in actinomycetes. And a little bit background of myself. So now I am in Denmark. Just as introduction, I'm in Technical University of Denmark. We are in this very international institute, Novo Nordisk Foundation Center for Biosustainability. It is located in Copenhagen area. And this is the nice building I'm in. And the section I come from is called New Bioactive Compounds section. So it is headed by Professor Sanya Pli and Professor Tim Weber. So what we are doing is we want to combine the power of biotechnology and informatic technology. So it's biotech, BT, and IT to try to find more antibiotics against primary gram-negative pathogens. And we have a nice setup flow from computational biology to wet lab. So the background is that, actually the drug shortage, as well as drug resistant pathogen is a huge problem for our human beings. And as the statistics saying that actually every year the mortality caused by drug resistant pathogen is more than cancer do. And the situation will get worse and worse because we simply don't have enough effective drugs, antibiotics. So that's why many people are saying that we need to find or develop more antibiotics for this kind of purpose. Saying is superbugs need drugs. And the thing is that we know that more than half of the antibiotics and anti-cancer drugs are coming from a bacteria called actinomycetes. So this picture shows very nice morphology of this organism. And during last century, we have a golden age of natural products-based antibiotics discovery. So it's basically from 1950s to 1960s. And suddenly after that period, so the speed and output of natural product-based drug discovery drops very fast. So the reason is that the lower hanging apple or fruit can be picked easier than those higher hanging apples. That's why now, because all those lower hanging apples have been picked, so we need some new tools, higher ladders to pick those higher hanging fruits. And what is those higher ladders? That is some new tools, some new technologies. And as you know that the sequencing becoming cheaper and cheaper. That's why now we have accumulated so many genome sequencing data, and this is stationary increasing. By this kind of nice data we have, now natural products discovery has been moved to bottom-up strategy instead of the classic top-down strategy. So simply saying that bottom-up is that we start from genetic information instead of we just start from the cultivation. And then we use modern technologies to do genome mining, and then based on the genome mining information, we do cluster cloning, then put the cluster into a cell factory, then use modern analytic method to finally get the expressed compound. And the compound can be some drug leads later on. If lucky enough, this kind of leads can be later on become drugs. So based on the genome mining information, we know that actually more than 80% of the gene clusters. So gene cluster means that, like, because these molecules is a very complicated pathway. So there involves multiple genes, and those genes normally are clustered. So we call it gene cluster. For example, one compound synthesize one compound normally needs 10, maybe 20 different genes cooperating, and then those genes clustered as a cluster. And based on the genome mining, we know that most of those clusters has not been assigned with any known compound. That means we have a high chance to have some new compounds out of these clusters. But the question is that how to get them from genomic information to final compounds. And I want to show you that this classic bottom-up way. So start from the strands. Now we have a high quality of genomes assembled, and then we can use some modern genome mining technology to get the genome information analyzed. And then from the genome information analysis, then we can have some cluster be predicted. And then this kind of big data, then we need to use laboratory work to do the evaluation. And now I want to give you some timescales, how these steps will be. For example, the first step from the strand to assembled genome maybe takes weeks to months, depends different like growth speed and the facility of genome sequencing. And then after you get in the genome information, assemble the genome, then you do genome mining, because nowadays the computational power becoming faster and faster. So this kind of analysis maybe takes just seconds to hours, maybe days. And then the last step is that from those predicted cluster information to the real compound, it takes years. And maybe you will never get the compound out. So now we see a big gap in between those computational biology to real laboratory work. There's a very huge gap in between. And because we can use computational biology to accumulate more and more data, so-called big data. But then we need to use our own hand to process this kind of big data. And that is just, we cannot do as faster as computer do nowadays. That's why I want to contribute in this area to develop some good tools, try to speed up this kind of processing. So this is old Chinese saying that if you want to do a job, you first need to have some good tools. And we know that before CRISPR, so doing genetic manipulation of actinomycetes is very, very hard. One or two reasons for this is that actinomycetes is a kind of gram-positive bacteria with a linear chromosome, and the genome is very high GC. I mean, very high is around more than maybe 70% of GC content. And if you are doing a molecular biology, you will know that if DNA fragment with 70% high GC content, even the PCR will be a problem, will be problematic. So that's why the classic method is very complicated, and I'm not going to give you the details. I just want you to know that it is time consuming and the efficiency is low, and it's a double crossover based method. And you know, like just kidding, I'm lazy, so I need something new to speed it up. And then when I step in this area, that is the later of 2013, and that is a very good time of CRISPR, and just CRISPR was emerging at that time. And this is how CRISPR works for genome editing. So simply saying is that first we have nucleus, for example, Cas9 or CPF1. So now we call it Cas12a. And then we give it a piece of guard RNA, so sgRNA. So sgRNA and this nucleus, they can form a complex. And this complex will kind of a scanner to scan the genome. And then if it finds a target, so the RNA, the spacer RNA in within this sgRNA will just pair to the target, complementary to the target DNA. Then this will cause the conformational change of the nucleus, and this nucleus will generate a double-strand break of the DNA target. Then after that double-strand break, so there will trigger the double-strand break repair pathway of the organisms. And during the DNA repairing, so we can have different outputs of genome editing. For example, we can use some defective non-homolog endogenin to have a random size deletion, or we can use non-homolog endogenin, I mean the complete non-homolog endogenin. Then we can just introduce some indels, small deletion, insertion, and substitution. Or we can provide the repair template. So we call it homolog direct repair. So that means we can do it as a cut and paste way. So it is a very precise and high fidelity genome editing. Now it comes to actinomycetes, and just like I just explained, so the magic of CRISPR-Cas9 genome editing is inside of the double-strand break repair. And I just mentioned there are two major ways. One is non-homolog endogenin, and that is the dominant way in eukaryotic cell, and the homologs recombination is used by most of the bacteria, because most of the bacteria is the non-homolog endogenin pathway is missing. So that will be a problem. So it's to be or not to be. So if you have a double-strand break, if you don't repair it, it will die. So that means it will force the bacteria to do the repair to survive. And this nice organism is a modern organism of actinomycetes called Streptomyces silicola. So this blue compound is Actinorhodin. So and we use this to have this proof of concept of CRISPR-Cas9 system. And this is the design of this system. So as you know that in actinomycetes, there are not many options of plasmid. There's not many choices. So that's why I put two major components into the same plasmid. And this is temperature sensitive plasmid. So temperature sensitive means that if you increase the incubation temperature of the target strain, so the plasmid, the repair count will be dysfunction and it will be killed, it will lost. That's a very good feature. That means you can repetitively use the same plasmid, the same backbone for multiple manipulations. And this is a shuttle plasmid, so it's E. coli and actinomycetes. I mean, because in actinomycetes, we normally use the conjugation to use E. coli as a donor to do the plasmid transformation. And this is the major important component, Cas9. And then here is the sgRNA part. So the Cas9 is under control of tetracycline. No, it's a type A, so it's thyrostreptone inducible promoter, and the sgRNA is under control of constitutive promoter, or ME. And just remember that I also have a sort of backdoor for some other components cloning to make the plasmid still all-in-one plasmid system. And just I mentioned that because most of the bacteria, it doesn't have the non-homolog adjoining pathway. But interesting enough, some of the bacteria has, and for example, the actinomycetes and the silicola, it has this Ku protein, and it has most of the function proteins of this ligase, but some parts is missing. So this is a ligase. So there's two major components for non-homolog adjoining pathway of bacteria. So this Ku protein and the ligase. And the ligase is a multifunctional protein, and it has at least 3 domains. So in silicola, we can see that Ku is there, and the primase is there, and there are so many DNA ligase floating somewhere, and some other parts are missing. So we saw that probably we can take advantage of this not perfect non-homolog adjoining pathway. So we just give it a try without providing repair template. That means the cell only can repair the double strand break by its internal mechanism, not by the repair templating. So here's the primary result. We can see that actually, if even we don't provide homologs recombination template, the CRISPR-Cas9 worked. So here I want to explain you a little bit. So as I said, that actinorhodin is blue, and in this color, so it can produce 2 types of pigments. One is blue compound, actinorhodin, and the other compound is red one, and the red one is not diffusible. That means if both red and blue are present, only the blue color can be seen, and the blue is diffusible. You can see that it can diffuse to the media. So you can see here, these 2 clones, it shows red. That means the blue compound, the target cluster has been inactivated. And also we can see from the spectrum, and it is actually, the blue compound is gone. And the blue compound, the pathway was the target that I use CRISPR-Cas9 to attack. And good, and we had some statistics of the efficiency. So the efficiency is pretty much varies from 3% to around 50%, because of different sgRNA gives different efficiency. That is common knowledge. And honestly, this efficiency is not good enough, only 50%. And next step is that we want to see why this efficiency is is not as we expected. And then we just randomly pick some clones for whole genome sequencing to see what's going on with the editing, if that editing is precisely introduced or just some side effect caused somewhere else. So we whole genome sequenced the 4 randomly picked clone. And funny thing is that we found just from these 4 different clones, we saw 4 different patterns. One is just, you see only one single base insertion in this cluster, and this one base pair insertion can just frame shift the whole cluster, and that's also no blue compound can be produced. And in this extreme case is that around 37,000 base pair has been deleted. And you know that the Actinorhodin cluster is only 25,000 base pair. And that means the whole cluster has been deleted and beyond that. So we were so excited by the time to see this kind of unusual result, because at the very beginning, we know that this non-homolog endogenic pathway is not complete. And that means this kind of becoming a part of competition. So when you have a double-strand break, so the cell want to survive, so it want to repair it. But in the meantime, there were many of the exonucleases in the cell, and they want to digest the chromosome because you have a double-strand break. And by that time, so it's kind of competition. So that's why like it will give you a random sized deletion. So theoretically, it can be in between of 2 essential genes, because if you destroy the essential gene, the cell will die. Even if you repair it, it still will be dead. So that will be a very good application, that because you just need one set of experiments, and if you picked enough clones, you can get a library of random sized deletion, and you can use this for different purposes. For example, to study the gene function, essential genes, and cluster identification, and cluster boundary identification, and so on and so on. So the next step is just I mentioned, because this non-homolog endogenic pathway is not complete, and we know that which part is defective. So we just want to repair this non-homolog endogenic pathway. And luckily, we by blasting we found 4 functional ligase homologs gene somewhere, and then we just synthesize it and put this cassette into the same plasmid. Just I mentioned that there's rejection enzyme sites that I can use for further things cloning. So still, this is one plasmid system, and I put this ligase expression cassette here, and to complement the non-homolog endogenic pathway. And then now we see a much better output. So after randomly picked 8 different clones, you see most of them is just a small insertion or deletion. So it's in there, just like a mammalian cell. And then you see that the editing efficiency is also increased a lot. So from 3% to 50%, now we can see around 70–80%. And that's a very good efficiency. And the next step is just I mentioned that there's the other way. Just use the editing template to do a homologous recombination repair, and that is paste and cut and paste repair. So for example, I just provide the left arm and the right arm of the target gene, and after a double-strand break, so the cell wants to survive, so it has to repair, and it can use this homologous recombination to repair it, and just to kill this target gene out, and to have seamless deletion. And now from here, just 10 randomly picked clones, I can get 10 randomly positive results. And this is around nearly 100% efficiency. And that is also consistent with other reports. So now we have a long story about this active CRISPR-Cas9. So we have different ways, different repair mechanisms. Then we can have different outputs. The next thing is that, you know, in some metabolic engineering, to have completely gene knockout is not a very good way. For instance, if you found later on this gene is not the one you expected, that means you had the wrong knockout, and then you need to start over. But then you cannot use the strain because the strain already has the permanent deletion. So that means that will be problematic. So here comes this knockdown system. That means the genetic information will be still intact. Just the gene expression has been knocking down. And luckily that this CRISPR system also can do this job. So if we just mutate the Cas9, because Cas9 only has 2 active sites for nuclease activity. If we mutate both, this D10 and H840, then it can still bind to DNA, bind to the sgRNA, and the complex still can bind to DNA, but there's no nuclease activity. Then this complex can be served as a roadblock. That means the DNA, the RNA polymerase cannot move if you have this block. That means the mRNA cannot be synthesized. That means the protein cannot be translated. That means that the pathway is sort of inactivated. So this is a nice system. So just as the RNAi, so people call it CRISPRI. So it's a CRISPR interference system. And we designed different sgRNA, both from the positive strand and the negative strand, target the same cluster, Actinorhodin cluster, the same gene, this ACT1F1. And we saw that just, you know, that if the pathway is inactivated, so the red compound can be seen, so it's red. And because I said that this whole system is based on temperature sensitive replicon, which means it is reversible. So if you just increase the temperature from 30 to 37, so see the red becoming blue again. So this is a reversible gene expression modulation system. Now I can give you a first summary of what I did for this CRISPR-Cas9 system. So we can just do a very simple comparison to the classic PCR targeting system. So the major advantage is that it is very quick, very fast. If you want to use this CRISPR-Cas9 system for knocking out a gene, it takes 3 weeks. And if you use the PCR targeting, probably it needs 2 months. And also it is highly efficient. Like it's if you use this homologous recombination repair, it will be around nearly 100%. So it is very good. But very soon, when we are using this CRISPR-Cas9 to do some other knocking out in other strains, for example, this Superbugs colonies, we had a problem. The problem is that we tried many times and we just cannot get the expected knockouts. So some background of this cluster. So this is a cluster for synthesize a compound called kirromycin. And as I just mentioned that most of the stromathids, it has linear chromosome. And linear chromosome, most of those linear chromosome has inverted repeat at the chromosome end. And this cluster, the kirromycin cluster, located in the end of the chromosome, inside of the inverted repeat, that means it has 2 copies. And 2 copies, we can just think that if we introduce a double strand break in one gene, because this has 2 copies, and it will give you 2 double strand break. So 2 double strand break means that you see a chromosome, linear chromosome with 2 double strand break. That means you have 3 repeats, and we saw that this must be too much for a bacteria to repair it in time precisely. That's why we go back to see after sequencing we observed that instead of seamless deletion of KRN gene, the entire cluster was lost. Both of the clusters has been deleted. So that's very strange, and we thought probably that's because of these 2 simultaneously introduced double strand break. So that is the motivation that behind. We want to establish something new, some new technologies that has more or less the same efficiency, but will not introduce double strand break. And this is CRISPR-based editing. So we named it CRISPR-BEST. So it's base editing system. And it has 2 types of base editors. One is from C to T, and the other one is from A to G. So long story short is that the same concept, we just fused the deaminase to CRISPR-Cas9 enzyme, but it's not active enzyme, and it's not also dead Cas9, but it's a Cas9 ligase. Ligase means that it can only introduce a nick in one strand, not both strands. And this is good for recruiting the DNA repair mechanism later on. That's why we use this ligase. So it has editing window, and it can do a lot of things. So this is a summary that what can base editors do. So you know that just from CG to CG pair to TA pair, or AT pair to GC pair, almost all kinds of amino acid exchange can happen. And in particular, that some amino acid can be introduced as a stop codon. And you know that stop codon is very useful. Like there are some possibilities to introduce stop codons. So stop codon means that if you give a stop codon early enough inside of a gene, that means this gene will not properly translated later on. That means the protein will not give you a full functional protein. And that is sort of equal to knockout. And later on, we did some deep characterization of this system. So we want to figure out the editing window, for instance, and which kind of genetic content will be the optimal genetic made up for base editing. And from the result, we can see that for C to T base editor. So the editing window is around 7 nucleotide. And for the A to G is around 6 nucleotide. And the best genetic content for CRISPR-based is T, the C behind the T. The same with the CRISPR-based is the A behind the T. Then we want to have some proof concept. So showcase one is we still use the model organisms of Silicala. Just I mentioned before that this blue compound, if you inactivated the blue compound, so the red can be seen. And the same, we can see that 4 different sgRNAs targeting this cluster. We see that the stop codon has been successfully introduced. So case 1 and this one is stop codon introduced, and number 2 is here. And we see that some other amino acid exchange happening, and that also will somehow disable this protein. And then this is the other gene inside of this class, also stop codon. And this is also the amino acid exchange. And in Silicala, we saw a very good output of this CRISPR-based system. Then we want to go back to solve our problem of kirromycin cluster, because I just mentioned that use CRISPR-Cas9, we can never get desired in-frame deletion of the KRN gene. And now we used CRISPR-Cas9, CRISPR-based system to introduce stop codon, and we see that it is very clear. First piece of evidence is that we can use PCR to get this fragment out. As you know that use CRISPR-Cas9 because the whole cluster has been lost. That means if you want to use PCR, there's no template. That means you can never get a PCR product. And here, the first piece of information is that I can get the PCR product and can get it perfectly single sequenced. So, and stop codon has been introduced. And the second piece of information is evidence is we use the bioactive guided evaluation, because this KRN gene, if we knock out, we inactivated it, we know that the kirromycin production yield will be much reduced compared to the wild type and the empty plasmid strain. And we know that kirromycin is active against bacillus. So we see the bacillus and we see that yes, the yield is reduced. And then the last step is, the last piece of evidence, you just use LCMS and we see that actually the compound yield is indeed reduced. And then we now know that CRISPR-based can do some task that CRISPR-Cas9 cannot. So it's good. And then we also had a very nice setup of pipeline for off-target effect evaluation, because you know for CRISPR-related genome editing, the first question or the first concern will be off-target effect, because this is a nucleus and you know DNA nucleus, and it also will maybe cut somewhere else. So this is the pipeline we set up for genome-wide off-target evaluation. So we use the Illumina sequencing to have the genome sequenced, and then we established algorithm to count the amino acid level changes across the chromosome. And interestingly enough is to see that we can observe some off-target effect, because this is a parental strain, and you can see this is just the background, and there's no very obvious increase from some bias. But from the C to T, we can clearly see that there are more C to T off target, and the same here. But good thing is for this A to G base editor, the CRISPR-BEST, there's no this kind of bias, and that means the A-best is more fidelity compared to C-best. And this is a deep analysis of this off-target, because we want to know if this off-target was happening inside of a gene, will cause some gene functions, or it just in some intergenetic area. That means maybe intergenetic area is not that important. And also we want to see that if this off-target, like for example, this C to T inversion can cause some other stop codon introduction or sort of thing. But we can see from here, actually, the number is reasonable low compared to CRISPR-Cas9, because CRISPR-Cas9 will somehow cause genetic rearrangement, and that is like huge piece lost and sort of thing. So long story short is CRISPR-BEST, at least based on the off-target evaluation, it is more fidelity than CRISPR-Cas9. Then the next step is that nowadays, if you want to do a metabolic engineering, during this one gene is normally not enough. So that means sometimes you need to simultaneously handling multiple genes. And for CRISPR system, there are many ways to do multiplexing. The most, let's say, straightforward way is to have multiple cassette of sgRNA. And multiple cassette, I mean, cassette means it has independent promoter, independent terminator. And just I mentioned before that for actinomycetes, that will not a good choice, not be a good choice, because there are not many promoters and terminators for choosing. And if you keep using the same set of promoter and terminator, that will cause a plasmid instability, because if you use the same, exactly the same piece of fragment, it will somehow have some self-homologous recombination, so the plasmid will be destroyed. And then we came up with a good idea. So we use this Cy4. So Cy4 is RNase, and it can specifically recognize this kind of RNA structure and have a cut. So that means if we just put them together, and in between we have this specific RNA sequence, so we can have sgRNA chain and just use one promoter to drive it. And after the transcript is there, so the Cy4, these RNAs can process the long RNA transcript to single piece of functional sgRNA. And then we also applied the Golden Gate assembly to do the assembly. So this is the 3-spacer 3 sgRNA case as a proof concept. So we target, one is the Actinorhodin, so the blue one and the red one, and then the other is this red one, and in between is the other famous compound CEA. So after that, you know that that the wild type, the parental strain, is blue, right? Because it has blue and red, and if blue is there, so red cannot be seen. But after this multiple targeting plasmid treat, we can see that there's no color at all. So it means the red is gone, blue is gone, and this one we didn't have color, but we need to use some other way to do the evaluation. For example, MS. But the take-home message here is the 3 spacer idea, or the Sci4 sgRNA self-processing idea worked. It's very good. That means we can simultaneously targeting multiple genes to do many other useful applications of metabolic engineering or even synthetic biology. So besides that, we also established sgRNA designer. So it's a Web 2, and you can just upload your own genome, and then you can just based on your own genome to customized design different target genes, different sgRNAs, and also it has full support now for CRISPR-based application. For example, you can click just show the CRISPR-based output, or just show only stop codons mutations, for example. The star means that from Q can be introduce a stop codon here. So it is very nice, and you're welcome to try this system. So now it goes to the second summary, that this is the classic CRISPR-Cas9, and then this is the overview of the toolkit I developed, and we have CRISPR-Cas9, CRISPR-I, and also the CRISPR-BEST, the base editing system. So now the other comparison is that if everything is optimal, to inactivate the gene using CRISPR-BEST takes only around 1.5 weeks. So the time has been even shorter. So this is the overview of the CRISPR toolkit for actinomycetes that I developed. So it has many different plasmid inside, but every single plasmid is one plasmid system. So it's very convenient. So from Cas9, we have 3 different plasmids, 3 different systems. One is that we can just take advantage of the native non-homework engineering. So this incomplete, this defective homework engineering pathway to generate a random size deletion library, or we can use this ligase to reconstitute the non-homework engineering pathway to have sort of precise genome editing tools to introduce small indels. And then if we provide the homology recombination template, so then we can use the cut and paste to have in-frame deletion or even knock-in, so insertion for some other purpose. And then if we use the Cas9 ligase plus the deaminase, so we can have CRISPR-BEST, CRISPR-C-BEST, and CRISPR-A-BEST to do the A to G converting, or C to T conversion. And the last part is that from the ligase, we can completely inactivate the Cas9 to make a dead Cas9. I mean, there's no nucleic activity at all. And using that, we can use this D-Cas9 sgRNA complex as a roadblock to just stop the movement of RNA polymerase. That means can have the knockdown phenomena of a gene. And because of this plasmid is a temperature sensitive plasmid, so it is a reversible system. And then we have some publications and we have some patents, and this system has been widely distributed worldwide. But this is not an end. It's just a start, because you will never know that there will be some even better tools coming out. And for sure, I believe that there are. So then comes to why I use CRISPR-BEST, the name. So the name is just, sort of for marketing. And best is just best for now. And later it will be even more good tools and even better, best too. And then we hope that using all those higher ladders, advanced technology, advanced genome editing tools, we can bring the curve back. So we can have another golden age of natural products-based drug discovery, and to find a more useful, effective drugs to address to the drug resistant problem that nowadays we are facing. The last slide is for some acknowledgement. First, of course, Novo Nordisk Foundation for providing money for all the research I conducted, and also the group member from this NBC new bioactive compound section at CFB, and especially our PI, Professor Sonia Pli and Professor Tina Weber and other group members. And now I'm happy to take any questions if you have some.
Dr. Tong, thank you for your wonderful presentation. I hope the audience has understood how much the CRISPR is very important to explore the hidden treasures. There is nothing but the natural products we can be able to generate from the various bacterial sources. You have given a lot of good information, and then interesting your findings about how that natural products are generated by using the CRISPR technology in actinomycetes. Our best wishes that you will get more findings in your new microbe by using your CRISPR tool. Okay. Just I want to ask some of the questions posted by our audience.
Yes.
Dr. Kavita Tirumurugan from VIT, she has asked how the CRISPR-based is precious to target the specific target gene without affecting the rest of the sequence.
That's a good question. So I will just go back to, basically I can just show you here. So the targeting, actually, that is the same as CRISPR-Cas9. Like, it is the same for all kinds of CRISPR-related application. So how to target a gene, that is defined by a piece of small RNA. So nowadays we call it sgRNA, so single guard RNA, and in single guard RNA we have a spacer, so just I showed here. So it is a 20 nucleotide in length, and that is coming from the target gene. And this piece of RNA will have like exactly complementary sequence to the target DNA. And that means if you put this system inside of your organism, and it will find the opposite strand of your target DNA and make complementary. That is how the targeting happening. And after they have this complementary, and that will cause some conformational change of Cas9 protein, and sort of activate the system. So either have a double strand break or single strand break. And then in the meantime, for the CRISPR-BEST, so because we have a deaminase, and this deaminase is fused to the Cas9, nuclease Cas9 protein, basically. And then it will be allocated in the same position. And so, and because one strand of the double strand break, the target, has been bound to RNA, and then the other strand will be free, and that the free strand will be accessible to the deaminase, and deaminase can do the job. For example, if accidentally here, for example, the inside of the editing window, if there is a T, and this inside if there's C, and this C will be converted to a T, C to T, and then in the meantime, it will give you a stop codon. So that's how it works.
Thank you, Dr. Tong. Another one question. Is this CRISPR-based system specific for actinomycetes, or it can be used for other system?
It is also a good question, and I can tell you that. So it should be a universal system. The only thing you need to do if you are going to use it for another system, the other systems, other different bacteria, other different organisms, you just need to know that if the plasmid, the replicon, is functional in your organisms or not. And that's one thing. And the other thing is that because later on you need to have the protein, functional protein, which means that if the codon is not optimal for your system, then you probably need to do some codon optimization. For example, you need to just based on your organisms codon usage table to redesign the protein sequence, the DNA sequence, and that's it. And besides that, I will not see any problems of using it, because eventually it is a protein, it is RNA, and RNA, protein, and DNA, they are just, every organism is the same. As long as you have a functional protein inside of your organisms, it should work.
Thank you, Dr. Tong. Is this technology can be used for the detection of COVID-19?
Sorry again?
Is this technology, this CRISPR technology,
yeah,
okay, can be used for the detection of COVID-19?
For degradation.
For detection.
Detection. Yes,
yeah.
yes. Good question. So that will be not the topic that I present, but I can give you some more information for that, especially for targeting. So for detecting, for detection. So nowadays, many people, many groups, researchers, they are using CRISPR CPF1. So maybe other people knowing as CPF1, but nowadays it has a new name, Cas12a. Just I had it here. So that is very interesting system. So it has here. So this one. So CPF1. So it also can give you a double strand break, but after this protein has been activated, so it has other activity. For example, it can cut single strand DNA, and then you can link many things to your single strand DNA for detection, right? So And then it can be used for detection of many different other things. If you can link your target to a single piece of olig, and this is for one application. And then the other application is other Cas protein. So it's ODAs coded C2C2. And that is RNA-guided RNA targeting system. That means you can use it to detect many other like RNA virus and many other things. So yes, it is also a very hot topic or hot area using CRISPR to do the detection, even for small molecules.
Okay, thank you,
Yes.
Thank you, Tong. Dr. Jebez, the time is there. Still a lot of questions are coming. So otherwise we can wind up the session.
So we can take one more question if you want, and questions can be posted directly to the professor, and we can get the reply from him and send it to the participants.
Okay,
yes,
okay.
I'm happy to do that, and I'm also happy.
Okay, Tong. Just we will wind up with another one question. Okay. How does CRISPR-Cas9 system in bacteria recognize bacteriophage or viral DNA as lethal target?
So if I understand correctly of the question is saying that why the
CRISPR,
so how this system was coming from the original bacterial immune system, you mean?
No. How does CRISPR-Cas9 system in bacteria recognize bacteriophage or viral DNA as lethal target?
That means, okay, so that's a little bit complicated. So first, if we want to use CRISPR-Cas9 system as a genome editing, then the thing is that you have to know that in your bacteria, there's no native CRISPR-Cas9 system, right? And because otherwise it will cause some so-called self-targeting. So because you know that at the very beginning, CRISPR-Cas9 system or CRISPR system is used as defense system or immune system for bacteria itself to stop the invasion of some alien DNA, some virus, right? And then so there's a very complicated mechanism for some bacteria to capture some virus or alien DNA fragment to integrate it into its own genome as like immune system as like spacer. And then but the bacteria itself will not trigger the expression of this spacer. Only if the second time the same piece of alien DNA or the same virus is going to infect the bacteria, so there will be some triggers to let the bacteria to respond to activate its expression of this part of the spacer, and then this spacer will form sgRNA, and then the sgRNA will form the complex with Cas9, and this Cas9 will go to attack the invader. So that's the immune system. And then later on is that people saw that, okay, if we can just stop the self-targeting, then we probably just use some other, like to use this system to other organisms without self-targeting, and we can provide some other repair mechanisms to repair the double-strand break. Even if like some bacteria, it can use as self-targeting, but in the meantime, you can just provide some repair template, for example, homologous recombination template, so it still can be used as a genome editing. So if that is the question you want, so maybe later if you don't like you want more, then I can privately give you more answers for that.
Oh, thank you, Tong. Thank you for your excellent presentation. So that we hope that the audience got a lot of understanding about the CRISPR tool for generating various compounds from microbial source. Okay.
Thank you, and the pleasure is mine.
And then those questions which has not been answered, the audience, you can mail to Dr. Tong for getting the explanation. Thank you for your participation. We hope that you got an excellent session. Thank you.
Thank you.
Thank you.